role of nutrients in the food and deficiency diseases. It will teach metabolism of nutrients in health and diseases. Thirdly, it will have different systems of the body and their functions with special reference to the digestion, absorption, transport, and uptake of the nutrients and the elimination of the waste products. Next, physiological changes also at different stages of life, and also it will help, it will improve them to understand the importance of therapeutic diet. So this is a short 1 year certificate course which teach them about the dietary control of different diseases also. So I hope these are the learning objectives behind this certificate course. So this is this these are the 4 units. Um Okay, let me a small introduction about unit also I'll give. Unit 1 speaks about food and food acceptance. So it will again teach students about the aspects of food, particular foods also, and it will also the students will be able to also define and learn different nutrients also. Next is they will be able to know the functions and deficiency of vitamins, particular vitamins, carbohydrates, fats, and also about the minerals. So, in short, this certificate again explains about the micronutrients to the students, and also third unit is regarding the basic food groups. So again, they will have knowledge about all the 5 food groups and also the meal patterns according to the states, like which foods we all different kinds of states consume, and also their pattern according to the states. Next is unit 4, last unit regarding the certificate course of this nutrition is, these is again they will teach you about the protein energy malnutrition, that is a particular deficiency disease, and again anemia and iodine deficiency disorders also, and nature causes, treatment and prevention too. Now this will bring an outcome in a student about related to deficiencies like protein, like any blood and iodine also. So whatever they will understand is the different deficiencies regarding particular uh, uh, vitamins or proteins. Next is they will also able to take the preventive measures, and also they will able to know about the different deficiencies and to how to avoid them, basically how to avoid the deficiency. So this was our fourth last unit for the certificate course. And again, my slide have prepared, but it is a bit very just a moment, I'll just get back to proper slide show. Yes, now next part is certificate course ka zoology department practical, that is 102. In this laboratory what we do is we teach them about the weights and measures. In short, just an introduction we give them. Again, we ask them to find out the calorific value, the nutritive value of foodstuffs also. In short, they are being taught how to calculate the BMI and how to calculate the energy. And also as see regarding these are 15 practicals, like methods and medium of cooking, various methods of cooking, like how advantageous and useful way you can cook more better ways. That is what we prefer in practicals. Mostly we teach them how to just theoretical part we teach them. Practically they might do it in their own houses. Few practicals which we are able to, dry practicals which we can show them in certificate course we can show them, like introduction of food guide hai, या फिर use of reference man, reference woman, like that. Or else we can ask them like to show the standardization of recipes, one serving recipe, like that. Such practicals are there. Another practical is identification of food items, small just advantageous practicals we have introduced in this certificate course, like original meal patterns, food preparations, and classifying recipes as good and moderate and poor. Then again, we teach about sources of specific nutrients, and also the demonstration of seasonal foods, locally available foods, and inexpensive quality foods. Then again, study of foodstuffs rich in more than one nutrient. and last is combination of foods. That's all for the certificate course, I suppose. And again, we have test for sugars, fructose, lactose, maltose. These kinds of qualitative tests of all the vitamins and calcium is there in this course. Then, in short, again, this practical work also shows various means of measurements to the students, also give them an idea about the fruits and vegetables and their measurements, and also how to analyze the blood, how to know the different meal patterns, and also how to test the different sugars, protein, calcium, and vitamins. We have another project work related to any subject. This year, I have given them nuts and oil seeds. I've given them various kinds of project, like you can do on any topic related to nutrition. So this time, I've I've just given them the option of nuts and oil seeds. So better progress has been shown by students. They have actually submitted the project of nuts and oil seeds. So syllabus for this course is Shri Lakshmi, which I also followed when I was new version. Hello.
Yes,
Yes,
ma'am.
ma'am. Yes, I hope I'm not speeding up now. Okay, I will explain now second syllabus for second year diploma program. Diploma in food science and nutrition. Yes. In this again we have in this sorry. Yes, syllabus for second year diploma program. So title of this subject is Diploma in Food Science and Nutrition. Yes.
मॅडम टायटल बदललेलं
Ha
आहे
ha.
त्याचं न्यूट्रिशन अँड डायटेटिक्स
Okay.
आहे हा फूड
Okay.
सायन्स नाही
Okay.
आता
Okay.
न्यूट्रिशन अँड डायटेटिक्स
अच्छा,
आहे
फूड ग्रुप्स
आणि
अँड मील्स
पेपरचं
आहे का?
नाव पण बदललेलं आहे D*101
ओके, स्टार वन
D*102
झिरो वन.
एल आहे एल लॅबरेटरीवर लॅबोरेटरीवर आणि नंतरच आहे ते
ओके.
पी
ओके.
डी
अच्छा.
स्टार वन
ओके.
झिरो
ओके. पी आहे. येस येस येस.
पी आहे
हो हो.
हा
पी फॉर प्रोजेक्ट,
ते करा आणि
पी फॉर
अर्ध
प्रॅक्टिकल
प्रॅक्टिकल
अँड
फक्त थोडे प्रॅक्टिकल फर्स्ट टर्मला आहे थोडे ते सेकंड टर्मला आहे
ओके. येस. ओके. Okay. Tama, this second year course, diploma course was implemented in 2021.
सेकंड इयर डिप्लोमा तो पण टायटल तेच आहे न्यूट्रिशन अँड डायटेटिक्स
Yes, yes, yes. So, okay, ma'am. Again, this is for second year DZT201, DZL202. Is it right, Sheikh ma'am? Is
येस
it right, Sheikh ma'am? See, I have
येस
just
येस
found out this paper. Yes, okay. So these are again paper number and the name of the code of the paper. This instruction is English. Pattern is semester pattern. This diploma course is semester pattern. Yes, we have two semester in this. Um, this is a diploma for zoology theory, nutritional planning, 201. Diploma zoology laboratory practical, 202. And diploma in project work is there, 203. That indicates theory, lab work, project work. Yes. Then it has two semesters. One is it has two units. Unit 1 and Unit 2 in first semester, TZT 201 nutritional planning. Uh, Unit 1 speaks about applied nutrition. Now, again, it helps students to know the various stages of life and how to take care of nutrition and their diet during these different stages of life. Like it starts from infancy to the old age, uh, and also to the physical activity of the different stages of life. So here we teach about all the kinds of RDA recommendations, all the kinds of developments happening in the growth, happening in the students, children, infant, and also the old age. So accordingly, they are taken care of. So we teach students here how to handle the situation when the ages, different ages, people come to you or come to us regarding the counseling. So this is how we take care of applying nutrition or applying the recommended dietary plans. Unit 2 says about deficiency disorders. Deficiency disorders like regarding vitamins, all thoroughly vitamins, fat soluble, water soluble, they are being taught here, and also the allowances and how to study the deficiency of all the vitamins are being carried in this unit 2. Like it includes zinc, copper, chromium, and also fluorine, and also the minerals which are important for your body. Semester 2 includes in this theory meal management. See, in meal management there are now in semester 2 it has 3 units. Basically family meal management, like how you're going to handle the full day meal pattern. Usually ladies have problem regarding how to manage every day ka meal, so including the nutrients and also the different variety of food patterns. Yes, so this family meal management teaches or make the student understand how small changes or small things can included in our day to day life. So this we make them learn regarding common Indian recipes plus with the different additions of different nutrients to it. Next unit, diet in genetic disorders. Now here it is thoda sa chemical chemistry and also science mixed in this nutrition background, ki how actually the chemical process takes place in our body and how that nutrient is important, and increase in any nutrient, how the digestion process changes or the chemical reaction takes place. So again here few of the theoretical part we teaches them, like phenylketonuria, galactosemia, or else fructose 1,6 biphosphate deficiency. These are all chemistry terms. Though we also take those detailed terms for the semester 2. Then lastly, we have nutritional biochemistry. It has again role of carbohydrates in nutrition, and its high and low levels, which affects your absorption, malabsorptions, digestion is being explained in this theoretical semester 2 pattern. Same way, it talks about proteins and also about the lipids. The same procedure we follow for the unit 3, roles of carbohydrates, protein, and lipids. So we have this. So like learning outcomes, I would love to tell key students will really get all the knowledge of vitamins, play each vitamin in the body, role of its, and also they will understand the sources of vitamins, basic sources. Most important outcome from this diploma course will be like the understand the genetic disorders, and also they will understand the diet in genetic disorders. So in both ways students, it will be helpful in respect with nutritional requirement at specific age or groups. They will be able to understand how the different people are there, or what the growth is, the different stages of life, and also the role of vitamins or the requirements of the body to maintain the balanced diet. And also in short, chemistry, when you include chemistry, I hope everyone is very familiar with this. It explains the breakdown and synthesis of each nutrient in our body. So that's what we have semester pattern 1 and 2. These were 5 units in 1 and 2 semester pattern. Then is again diploma for zoological laboratory practical, that is semester 1 202. We have a few practicals regarding the healthy cooking practices related to your theory part, like how it can be cooked, how it can be stored, how it can be prepared, and with specific nutrients included, not included, like that ways. And semester 2 practical, we have preparation of nutritive value, basic preparations. Then like milk and milk products, how the basic preparation takes place. You can add this milk to the make your nutrition or make your thali look more nutritious and also have more nutrition uh, in your basic balanced diet. Then again meat is there, eggs is there, soup is there. So this is what semester 2 practical we do. Meal planning of different age groups. Again, we do the balanced diet for pregnant women, lactating women, preschool child. This we don't go practically actually, but we, yes, we have theoretical part. We have how to make diet for these people, lactating हो या preschool child हो या school going child हो, lunch box हो बच्चों का या फिर adolescents हो, senior citizens at a home level. We teach, we plan the and prepare the diet for these different age groups. Yes, and yes, not the least, physical fitness of individual and nutrition assessment. So this is all included in semester 2 practical. So again, these are, there is a project DZP 203. One project is there again for 50 marks. So this they here also we have given them options of making any project. I see most of the students doing vitamins. Vitamins is what they have understood very well. So they keep on doing a project of vitamins. So at least some kind of information they have been given regarding this diploma courses. So they actually understand, yes, this is what is important, and both ways, like physiological needs, like of different stages of life, and as I told you, like we prepare and plan their own nutritional food recipes also. And the learning outcomes will be again different meal patterns, which is very important nowadays. People really require different colorful, but with nutrition is one another very important point. Not only colorful food you should have, but it should be based on very good nutrition. So this is what different meal patterns we teach. Then we have dietary importance of fruits and vegetables in their own ways, and this is what they will learn regarding the nutritive sources of their meals. I hope I have not hurried so much, but thank you. Thank you so much. Yes. Any? Uh,
But yeah, ma'am.
yes. Yes.
Hello.
Yes. Yes.
Yeah, ma'am. In the topic of meal management,
Huh.
I think nutritional planning, do you have any specific charts that shows the proper proportions of that of different age groups so that we can show
Yes.
to
Yes.
students?
Okay. Okay. Different.
So if you get time, then please give. Yeah,
Okay.
means
Uh, right now though I have to search for it. I will
any
have it. I
chart
have
regarding
actually shared it with my students. I have shared it, but then I will just moment I will just search it and I'll put it in front of you if I get it. But right now I don't have. I'll manage it. I'll next time na I'll pakka show all the charts also if possible.
okay,
Anytime
okay.
if you arrange it I
Yeah,
will
means
show.
only that chart can means we can. Okay,
Yes
ma'am.
ma'am.
Yeah. This will help for easy understanding that actually how we have to keep the proportion chart in
Yes
front
ma'am.
of
Yes
them
ma'am.
if you
Diploma.
supply it to them.
Okay. For
Yeah.
the students, you are saying for the apna, the student purpose, or now you want to see?
No,
I
if
have.
you're not having now, it's okay. You can send it to me
Okay.
afterwards.
Okay. Yes. Sure. Yes. Like right from infancy, we have planning of these people diet, planning regarding small age, infancy group 2. The old age, yes. Anything related to any other queries?
And ma'am, do you want to suggest any reference book so that we can buy it here that we don't have over here in our department
Yes,
for
yes.
teaching all this syllabus? You can suggest us.
Yes, reference books. Yes,
Apart
these
from
are.
this,
I hope you're able to see this. I've made it. Other
yeah,
from
yeah,
this, when we were studying, we actually referred only Sri Lakshmi because it was very helpful, and she has already given
yeah,
many editions, and so it is
that
enough.
was
Means
easy.
more than
Yeah.
enough because it keeps repeating. Otherwise, it's very nice. All books are good. Even Gopalan is good.
Okay, okay.
Swaminathan M is also very nice. Essential of Foods and Nutrition. That is also very nice.
Chalega. Means enough. No need to buy more. We
Yeah.
have editions of this
Okay.
there.
Thank you. Yes.
Yeah.
Yes.
Thank you, ma'am.
Actually, yes. Yes. Thank you so much. And thank you so much. Okay.
Okay. Thank you, ma'am.
Uh,
Yes. Thank you.
Dada
Thank you
sir,
so much.
करतो वोट ऑफ थँक्स करतो. Uh, it is my immense pleasure to propose vote of thanks for this session. So
Okay.
I'm thankful to the for this session resource person, Mrs. Indira Gauri Barge ma'am, nutritionist from Pune.
Thank
Thankful
you.
to giving valuable information on the revised syllabus of first year and second year diploma in nutrition and dietetics. Thank you, madam. I'm also thankful to
Yes.
the head of the department and vice principal of the institute, Dr. V.Y. Deshpande sir, to support to organize this program. And I'm also thankful to the principal of the institute, Dr.
Okay.
Vijay Jadhav sir. Thank you very much, and thank you all the faculty.
Thank you. Thank you so very much. Thank you.
Okay. Uh, Now we move to the next section for the faculty development program of advanced diploma in biomedical instrumentation. For this session, our resource person is the Dr. Swapnil Desai. Within 2 minutes, within 5 minutes, he will join, and we will start the next session for the advanced diploma in biomedical instrumentation within 5 minutes. Okay. Welcome, sir. So we start the our next session for the faculty development program in biomedical instrumentation. So for this session, the resource person is the Dr. Swapnil Desai, sir. He is working as a research associate in Health Accurate Diagnostics Center in Pune. His qualification is MSc and PhD in biotechnology. He completed his PhD from Savitribai Phule University, Pune. He also working in National Institute of Virology, Pune, and he published more than 10 research papers in national and international journals. And he also qualified the CSR NET GRF with 95 rank. So with the short introduction, I request to the today's resource person, Dr. Swapnil Desai, sir, to deliver his talk. Over to you, sir. Mic unmute करा. Mic unmute.
Hello.
Okay, sir.
Am I audible?
Yes, sir. Yes, sir.
Okay. So thank you very much for having me. So should I start directly, or is there any other introduction I have to give?
Yes, sir.
Should I start directly, or there is one more means kind of introduction? I have to
Only faculty is there.
Okay.
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 गूगल से आएगा डायरेक्ट हाँ शेयर करो
नहीं
डोंट ऐड काय म्हणते डिफरंट विंडोज इन दॅट
लगेगा.
थांब स्टॉप प्रेझेंटिंग म्हणून दुसऱ्या ह्याच्यामध्ये राहू दुसरं क्लिक विंडो टॅप टॅप करताय का
मगाशी विंडोज
करून बघ क्लिक करू
सर एंटायर स्क्रीन करा शेअर करताना.
ओके ओके.
हा प्रेझेंट नाव मधून.
क्लिक कर शेअर म्हणून
स्क्रीन प्रेझेंट झाली. पीपीटी ओपन करा फक्त.
मला दिसत नाही. दिसतंय का आता?
नाही अजून.
दिसते का विषय
आता? हॅलो.
नाही नाही
दिसतंय
नाही 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 दिसत.
नाही दिसत सर.
दिसते स्क्रीन सर.
नाही त्या ह्याच्यात ना त्याच्या आपल्या हिला विंडोच्या अलीकडे बुक हां क्रोमवरती जा त्याच्यात कुठल्या ऑप्शन पलीकडच्या ना
इथं पाटील सर
इथे
इथं ऑप्शन आहे प्रेझेंट नाव म्हणून. अप एरो आहे तिथं. तुम्ही फक्त स्क्रीन प्रेझेंट झालेली एंटायर स्क्रीन पीपीटी ओपन करा.
पीपीटी ओपन केली पण दिसत नाही कधी.
ऑप्शन पहिला कुठला यूज करा
प्रेझेंट नावचं कुठे गेलं मगाशी होतं
ते क्लोज करावं लागेल स्टॉप करा
स्टॉप करा 1 मिनिट, मग परत हे करा.
त्याच्यामध्ये कुठला ऑप्शन घेऊ म्हणून विचारा शेअरिंगचा हा त्यामध्ये विंडोज का कुठला हा
हा ऍक्च्युली एंटायर स्क्रीन प्रेझेंट करायचं ना
पीपीटी ओपन करून ठेवा आणि शेअर स्क्रीन केल्यानंतर पीपीटी ला सिलेक्ट करा.
एंटायर स्क्रीन ओके इथनं करायला लागेल. आता
एंटायर स्क्रीन झालेली आहे, पण हे तुमची पीपीटी
एक मिनिट आता येते का बघा.
हा आता पीपीटी ओपन करा. हा
आलं
ओके.
Okay. Good afternoon, everyone. So, I am going to present a small presentation on tools and techniques in molecular biology. Am I audible?
Yes sir, yes sir.
Okay. Just let me know if I am not audible or is there any problem. Okay. So basically, I'm going to give a small talk on the tools and techniques in molecular biology. Though the title is tools and techniques in molecular biology, but the techniques which I am going to discuss now, they are also useful in the diagnostics. So I will start now. Targets in the biological specimens means generally when we work on the any biological systems, most probably we are interested in the nucleic acids or enzymes and proteins. And no doubt, though they are abundant in the cells, they are not visible directly to the naked eyes. So what we do generally, or in research or in diagnostic, generally what we do, we try to amplify the signals. So how to amplify the signal? It is same like what we use. Suppose we are speaking on the microphone and we have to amplify the signal. We use the loudspeakers so that the small signal can get converted into the louder one. The same kind of technique is used here. So for the amplification of signals, we can use the fluorophores, we can use the dye, or we can use the radio label isotopes. So what are the fluorophores? Fluorophores are the compounds which on excite, they absorb one wavelength and they absorb one kind of means of 1 nanometer of wavelength and they emit another wavelength means the total visible system you've got. It starts with the violet color and ends in the with the red color. So fluorophores, what they do, they absorb suppose violet color and they will emit green or red color. So from the they absorb the wavelength of shorter wavelength and they emit with the longer wavelength, longer wavelengths. So this kind of fluorophores we can use. Also we can use the direct dyes. Dyes like for the proteins we can use the CBBR, chromazy brilliant blue. And for the DNA and RNA we can use ETBR means ethidium bromide or cyber green. So all these dyes and fluorophores we will discuss it later on in detail. Just I want to mention here only the fluorophores and dyes we use. Even PCR also is kind of technique which upscales the DNA and RNA. Means in few hours we get around millions or billions copies of DNA or the RNA. So PCR means polymerase chain reaction. This is the technique which revolutionized the total molecular biology field. So before PCR, it was hard to amplify or to see the genomes or the transcriptome of the cells or the living organisms. But PCR helped us for the detection and amplification of the target genes or the specific gene of interest. So Carey Mullis is the inventor of the PCR machine, and this invention was kind of serendipity, but that doesn't mean he was lucky in the he was just lucky. That doesn't mean he was lucky. It means that 24/7 he was thinking about the PCR or related things, and that's why suddenly he cracked the idea of PCR machine. So the interesting story behind it is he was traveling towards his home through the mountainous road in the night, and he suddenly got the idea of the PCR machine. And like all other great inventions, at the beginning, nobody believed in him. So when he tried to publish the paper on the same in 1985, the Nature Publishing Group, he rejected his paper. But later on, it showed that it was the important technique, and in 1993 he was awarded with the Nobel Prize. So what is the PCR? As name suggests, it is the polymerase chain reaction. Polymerase is the enzyme which goes on adding nucleotides from 5' to 3' end. Hence it is called as chain reaction. Why chain reaction? Because there are multiple cycles which we can set in the protocol, around 40 cycles, 30 to 40 cycles. So in this 30 to 40 cycles, millions and billions copies are generated. Am I audible?
Yes, sir. You are audible.
Okay. Okay. So millions and billions copies are generated in 30 to 40 cycles and in 1 to 2 hours. So what is the components of the PCR reaction? For the PCR reaction, we need the template. Template means the target gene or the gene of our interest, which we want to amplify. So for this, secondly, we need the polymerase. Suppose we are working, if we want to work on the DNA, we need the DNA polymerase. And if we want to work on the RNA, we need the reverse transcriptase, because directly PCR machine can't amplify the RNA. So we have to convert it first from the RNA to DNA, and it is possible with the reverse transcriptase. So these enzymes, they are isolated from the biological systems only. Also, they can be cloned, and we can use the recombinant enzymes also. But in the beginning, they were also isolated from the organisms. Third, we need the primers. So what are the primers? Primers means oligonucleotides, around 18 to 20 bases, which are used for the amplification, because DNA polymerase, it directly can't sense the template or start point. So we have to give something to amplify. So that's why primers are needed for the DNA polymerase. So these primers we can synthesize by using oligonucleotide synthesizers. So basically we order them. So how they are designed? Primers, for the designing of the primers, we need the sequence. If we want to work on the known virus or the bacteria, then already, hello, hello,
Yes, sir.
am I audible? Because
Yes,
I'm
sir. Yes.
thinking in
Yes, sir. You
between
are audible.
I'm losing connection.
No, sir. Yes, you
Okay,
are audible.
okay. So primers, for the designing of the primers, we need the specific sequence, genome sequence. And suppose we are totally working on the total new organism, then we can design the random universal primers because we don't know the sequence. But if we want to work on the known organism or the known virus, there are readily available genome sequences in the PubMed. So we can download the total full genome sequences from the PubMed, and then we can design the primers. Also, there are softwares are available for the design of the primers. Fourth one, we need the nuclear DNTPs. DNTPs means deoxynucleotides. Deoxynucleotides means the oxygen is missing, and oxygen is missing at the 2' end. So they are called deoxynucleotides. And for the activation or for the efficiency or to work efficiently, there is a need of the buffer for the enzymes. So buffers are also designed with the specific pH at which these enzymes can work. So steps involved in the PCR. There are 3 steps which are involved in the PCR protocol. First one is the denaturation, second one is the annealing, and third one is the extension. So what happens in the beginning when we add all the components in the tube? Actually, one more thing is like all the PCR systems work in vitro, means we use the small tubes or the strips or the plates, 96 well plates, for the amplification of these products. So this amplification is also called as in vitro amplification. And the PCR machine also called as a thermocycler. As name suggests, again, thermocycler or thermal cycler means thermals means related to the temperature, and cycle means we give the rotations, we give the cycles so that it continuously can work in the rotation. And why the thermal? Because in denaturation, we use the 95 degrees Celsius. For the annealing, we use the temperature around 50 to 60 degrees Celsius. And for the extension, we use the temperature around 68 to 72 degrees Celsius. So first we will speak about the denaturation. So the template or the strand or the DNA in which we are interested is basically double stranded. So double stranded DNA is not accessible for the amplification or for the propagation. So there is the need of denaturation. So first at the 95 degrees Celsius, you can see here the 2 strands get separated from each other. Then from the 95 degrees Celsius, the temperature is lowered down around for 50 to 60 degrees Celsius, so that primers can anneal to the complementary strands. So basically in the conventional PCR, we use 2 kinds of primers. One is the forward primer and another is the reverse primer. So forward primers work from the 5' to 3' on the complementary side, And reverse primers also work in the 5' to 3' direction again, but in the reverse direction. So for the conventional PCR, we need the forward primer as well as the reverse primer. So the primers get annealed with the complementary DNA, and the third step comes the extension or the amplifications. Once the primers get attached, now the polymerase starts the adding of new DNTPs. So it starts extension, and again and always it will start from 5' to 3' direction only, because at the 3' there is a hydroxyl group which is available for the phosphodiester bond from the 5' sides of the phosphate groups. So I will explain again in detail. And these polymerases are derived from the generally bacteria which grow at higher temperatures. So these bacteria are generally thermophilic bacteria. And the reason we use the DNA polymerase of the thermophilic bacteria is they can sustain for the higher temperatures, because in the denaturation we use the 95 degrees Celsius temperature. So at that temperature the polymerase or the enzyme should be sustainable. Hence we use the polymerases from the thermophilic bacteria. And the amplification is generally from, what we can say, suppose we can give the 30 cycles or 40 cycles. So when we give the 30 cycles in the protocol, it means that 2 raised to 30 means 2 are what multiplied 30 times, means 2 raised to 30 copies are generated in the 30 cycles. Suppose we give the 40 cycles protocol, then 2 raised to 40 copies are generated. So within 1 or 2 hours, around billions of copies are generated in the PCR machine. So second technique is the agarose gel for electrophoresis. So this agarose gel electrophoresis we use for the visualization of the DNA or the RNA. So the source of the DNA can be the PCR amplified product, which we have seen in the PCR technique, or the DNA or RNA can be extracted directly from the biological system. Suppose we want to isolate the plasmid from the bacteria, and if we extracted the plasmids, we can directly run on the agarose gel. Or suppose we are working on the tissue culture system and we want to check the total cellular RNA and its integrity. So we can extract the RNA from the total cells from the tissue culture, and we can run it on the agarose gel to visualize it. So how is the agarose gel electrophoresis works? Again, as name suggested, agarose is the kind of powder form, but it is derived from the seaweed. So it is derived from the seaweed. So how it is prepared? Agarose gel. Gel is basically same like the jelly chocolate, but it is not the edible. So agarose powder is weighed. So according to our requirement of the concentration, suppose we are running the DNA in the agarose gel, we can use the 1% gel. If we want to run the RNA, then it needs the 2% gel. As we increase the percentage, the size of the pores goes on decreasing. So for the RNA, our RNAs are the, as compared to the DNA, they are smaller. So to visualize better, we use the higher concentration of agarose gel, and for the DNA we can use the 1% gel. So how it is prepared? So agarose gel, so agarose is in the powder form. We dissolve it in the TAE buffer, then we boiled it in the microwave oven. After sufficient boiling, we allowed to cool it. When the temperature becomes bearable, we add the ETBR. So ETBR is the dye. You can see here. In the B image, you can see the DNA bands showing glow. So when you look under the gel dog system, you can visualize the DNA in the same pattern. So how ethidium bromide works? Basically, ethidium bromide is the intercalating agents. Intercalating agents means within the double strand, the hydrogen bonds are there. So ethidium bromide goes there and sits in between the hydrogen bonds. Hence, it is called as intercalating agent. So once we add the ETBR in the what solution, then we allowed it to cool in the casting tray. Once it gets the cooled, it becomes the jelly-like slab. Also during the cooling, we keep the tooth comb so that it generates the well in between the inside the agarose gel. So here you can see in the image A, you can see the tip, and from the tip it is low DNA, means it is the schematic representation, but we can imagine DNA or the RNA is getting loaded. So additionally, there is a loading dye which is in blue color. Actually, there is no connection of this loading dye with the actual visualization of the DNA. Just loading dye is added just to know how much run is completed on the gel, because again the DNA with the naked eye is not visible. Even with the ETBR, after addition of the ETBR also, the DNA or the RNA is not visible to the naked eye. So the loading die is just used to check how much run is get completed. And what is the principle behind the running of the what we can say DNA or RNA? Generally, DNA or the RNA is the negative in charge, and it is due to the phosphate group present on the DNA and the RNA. So basically, when you run, so in the tank buffer, sorry, in the tank, we pour the gel. Also with additionally, we put the tank buffer in it. So tank buffer is the TAE buffer, which is prepared of tris, acetic acid, and EDTA. So ions are present already in the buffer system. So when we pass the current, the negative charged DNA runs towards the positive charged anode, and this is totally based on the charge. So while running from the negative charge to the positive, it also gets separated according to the molecular weight. It means The smaller fragments of DNA will run faster, will come towards the positive strand in less time as compared to the larger strands or the large DNA fragments or the RNA fragments. So once the run is complete, you can use the UV illuminator or the gel dog system to visualize the DNA. So actually the DNA is or the RNA looks like the picture from the B, means B is showing the correct image of the DNA or the RNA when we look under the gel dog system. So one more additional use of the agar gel electrophoresis is in the diagnostic. Suppose the patient is positive or negative, we can only see when the gel is run and after visualization on the UV illuminator or the gel dog system. So in the diagnostic, how it is used, suppose we want to find out the specific infection of the virus or the bacteria. So we design the primers accordingly. We run the PCR, and after running the PCR, we try to find out whether patient is positive or the negative by running the agar gel electrophoresis. So in the end, if the patient is positive, we can find out the bands of the specific bacteria or the virus. The third system is the third technique is the sequencing. Again, it is the kind of revolution in the molecular field, because before that it wasn't possible to find out the phylogenetic link or even the genomes and the specific genes of the organisms. So once when we started the sequencing, or when we started the use of sequencing, the research become more easier. So how it started? There was a scientist named Frederick Sanger in 1977. So he developed the technique also known as the Sanger sequencing, and it is the oldest and widely used sequencing method. Now the next generation sequencing has come, but it is not the common and cost effective. So generally in most of the labs, Sanger sequencing is used. So at the beginning, when the Frederick Sanger started the sequencing or started this technique, he used radio label primers to find out the sequences. But nowadays, few developments has come, and now the sequencing has become the automated. Before that, at the time of the Sanger, the sequencing was the manual, and lots of gel has to be run to find out the sequence. And the automated sequencing shipped the radio label primers to the fluorophores. So the primers can be used with the fluorophores. So this automation was developed by the Hood and Smith. So what is the principle behind the sequencing? Here you can see the difference between the DNTPs and DDNTPs. So how the polymerization starts in the PCR reaction? Just see the 3' OH or the hydroxyl group in the DNTP, in the schematic representation of the DNTP. So this 3' OH is involved in the phosphodiester bond of the next nucleotides and the chain reaction continues. So to terminate this chain reaction, Sanger developed the dideoxynucleotides, that is the DDNTPs. So at the 3' end in the DDNTPs, you will find oxygen is missing. So oxygen is missing, that means the phosphodiester bond will not form and the polymerization will not occur. So generally in the sequencing, we use the DDNTPs along with the DNTPs. So every time when the DDNTPs is get incorporated into the sequencing, the reactions get terminated. So in the end, thousands of overlapping fragments are formed, which are detected later on by using the analyzer and the detection system. So this is how the sequencing works. So for the sequencing, components needed are the template. Again, we can use the DNA. Again, if you want to sequence the RNA, we have to convert it first into the DNA. Then only we can sequence in the Sanger sequencing. But in the NGS or the next generation sequencing, we can directly sequence the RNA. But for the Sanger sequencing, there is a need to convert RNA into the DNA. And all other components like DNA polymerase, DNTPs and DDNTPs or the buffer, there is a readily used solution is available which is called as TRRM, means termination ready reaction mixture, means everything from the DNA polymerase, DNTPs, DDNTPs and the buffers and additional uh, uh, what we can say, the additional ions, positive ions, which can enhance the uh, polymerase activities like MgCl2 are already added in the ready to use mixture, which is called as the TRRM, means termination ready reaction mixture. So basically sequencing occurs in 3 steps. In the first steps, we use the asymmetric PCR. Why it is called asymmetric PCR? Because like conventional PCR, we don't use forward primer and reverse primer in the same tube. Instead, for the asymmetric PCR or for the sequencing, we use the forward primer in the separate tube and the reverse primer in the separate tube. So basically for the single sequencing, 2 reactions goes. And as you can see, DDNTPs are generally DDATP, DDCTP, DDTTP or the DDGTP. Means this is the derivative of adenosine, cytosine, thymine or the guanine. So from all these, one more oxygen is removed, which were already present at the 3'. So this mixture along with the fluorophores is used in the sequencing. So whenever the different fragments or different threads are amplified, they will be terminated randomly where the DDNTPs are incorporated. So once the asymmetric PCR gets completed, the components are removed by the post-cycle purification, where we use the EDTA, we use the sodium acetate or the 70% ethanol to remove all the components or all the reminiscent which were there in the end of the asymmetric PCR. So in the post-cycle purification, we try to extract only the DNA fragments which are in which we are interested. So once we get the fragments, we put it into the automation machine or the analyzer. So analyzer, how it works? Generally, all the DDNTPs are with the different colors. Suppose here you can see the A, C, G, T. All the four colors are with the different, all the four nucleotides are with the different fluorophores or the different colors. So what happens in the sequencer or the sequencing machine or the analyzer? What we have seen in the conventional PCR, after the completion of the PCR, we use the agar gel. Here, instead of agar gel, we use the capillaries, different capillaries which are embedded with the polymers. So these polymers act as the same, they act as similar like the agar gel. So in the agar gel, different sieves or different pores are formed through which the DNA moves. In the sequencer, in the sequencing machine, capillaries are there along with the different fragments of the DNA are different fragments of DNA travel. So while traveling, even the single nucleotide is get detected. So how it is get detected? There is the laser beam which continuously uh, throws the laser on the sequencing threads. So when the DDNTPs come, they get excited because they are already with the fluorophores. So they get excited and the signal is generated. So this signal is detected by the detector and it is analyzed by the software system. And you can see in the B picture, the different peaks are seen. So all the peaks are, so the peaks are of the different nucleotides and the sequence you can see above. This is the schematic representation. So this is how the sequencing is done or the sequence is found out. So at the same time, we can use 96 different samples for the sequencing. And the peak or the diagram which is showing in the B picture, it is also called as the electrophoregram or the chromatogram, which is generated by the software. So this generated file you can check by using the different bioinformatic tools like the Mega 6 or the codon code aligner. Okay, so the next technique which we use in the diagnostic as well as So this is sequencing. In the sequencing, it is again for the total full genome sequence of the viruses or the bacteria or the humans or the fungus, anything, any living system. So we can sequence or we can collect the data or the genomic data of the any living system by using this sequencing machine. So these are important for the research, in the sense, suppose we have to develop something antimicrobial or the antibacterial, sorry, antiviral drug, or we have to block the specific protein. So for that, the sequences are necessary or the important. So after the sequencing, there is one more technique used in the diagnosis, is the real-time PCR. So this is nowadays you might be hearing a lot about the RT-PCR, because in the COVID-19 diagnostic we use the real-time PCR. And why it is called as the real-time PCR? Because like the conventional PCR, means it is unlike conventional PCR. Why? Because after the completion of the conventional PCR, we run the products on the gel, and after that only we can visualize the amplified products. It means that it is the laborious or it is a time consuming method. In the real time PCR, there is no need of the agar gel. We can see the amplification from the beginning, means from the first cycle, from the fifth cycle, suppose from the 10th cycle, we can directly see the amplification going on. Hence it is called as the real time. Real time is like you are watching live, amplification. So it is called as the real time. And as compared to the conventional, it is highly sensitive, is highly reproducible. The sensitivity is, means, suppose 10 copies of the virus in the blood or in the cell, we can detect. It is that much sensitive. Hence the viruses like airborne viruses or the pathogens, it is very important to get the early detection. So for the early detection, real-time PCR is the best technique we can use in the diagnosis. So there are two types of the real-time PCR. One is the comparative and one is the absolute. So comparative quantification is like the relative, means we put something as a standard and then we compare our target gene or the target DNA. In the absolute quantification, we can determine the copy number. So in the viruses, like in the diagnostic, like HBV, HCV, or the hepatitis E, or even in the uh, coronaviruses, we can actually find out the copy number in the blood or the copy number in the cells. So how it works? So for the real-time PCR, we can use DNA or the RNA as a template. So again, if we are using the RNA, it is the two-step or the one-step. Similar as we discussed in the PCR system, RNA is first converted into the cDNA or the complementary DNA by the reverse transcriptase. So when we use the two-step RT-PCR, the separate complementary DNA synthesis is done, and then we use the real-time PCR. And in one-step RT-PCR, within the single protocol, RNA is converted into the DNA, and then DNA is detected with the probes or the cybergreen. Cybergreen is the similar like agarose gel, like ATBR. It is the dye which sits to the minor groove of the double-stranded DNA. So it can bind non-specifically, similar as like ethidium bromide. And one more chemistry is there, which is the Tachman chemistry or the probes. So in case of DNA or the RNA, you can use cyber technique or the Tachman chemistry, which is the probes. So what are the probes? So probes are nothing but the complementary sequence or complementary oligonucleotides which are with the fluorophores and the quencher. So here you can see when we use the cyber green, it is non-specifically getting attached with the minor groove. But probes, they are highly sequence specific, so they will not get attached randomly. Hence, the Tachman chemistry or the Tachman probes are more sensitive, and in the diagnostic, mostly the Tachman chemistry is used. So cybergreen, where it is used in the relative quantification, means like suppose we are comparing the control cells with the infected cells. So we can give the, in the tissue culture system, we can give the infection of the infection to the cells by the bacteria or the virus or any other pathogen. And then we can study the gene upregulations or the down regulations with comparison with the control genes. So in that case, the cybergreen technique can be used. Also, as compared to the Tachman probe or the Tachman chemistry, cybergreen is the cost effective. Hence, cybergreen can be used in the real-time PCR system. So, how the probes work or how the Tachman chemistry works. Again, for the real-time PCR, we need the template, means gene of our interest. So what happens? The Tachman probes are actual complementary to these sequences. So they go and attach to their specific sites, which will be the complementary to the actual target gene. And PCR primers are the different. They also will go and attach to the complementary sequences, similar like what we have seen in the conventional PCR. But the DNA polymerase, what we are using here, it will go on adding at the 3' end the new nucleotides or the new DNTPs, and when it reaches to the Tachman probe, it has the 5' exonuclease activity. So exonuclease activity, it means it will cleave the fluorophore from there. Once the fluorophore is get cleaved, you can see in the image the fluorophore, once it gets separated from the quencher, it shows the fluorescence. So along with the quencher, it fluorescence is get shadowed, or its fluorescence it get inhibited when the amplification start and the DNA polymerase cleaves the fluorophores. It starts showing the fluorescence and it continues the polymerization. So new strand will form there, but the fluorophores and the quencher will be separated from the new strand. So this fluorescence is now get detected by the again detectors and the analyzer, similar like what we have seen in the sequencing. So here I am giving you the different types of fluorophores which are used in the real-time PCR. Here you can see the cyber green FAM. So they have the one wavelength with excitation and one wavelength with the emission. So once it gets the excited, they release the energy in the form of fluorescence, and this fluorescence is get detected by the analyzer. On the left hand side, I'm showing you the actual image of the what I can say it is from the COVID-19 diagnostic. So the amplification you can see it is in the sigmoid curve, and the genes which we are using the fluorophores which we are using are the FAM, Hex and the ROX. Out of this, the FAM and Hex, these two fluorophores are used with the viral gene, and ROX is used for the human gene which is the RNSP. So here in the target system you can see the Hex is for the N gene of the virus, ROX is the RNSP gene for the human. Further I will explain is this. So on the left hand side upper image is with the only FAM. So all these what you can see are the sigmoid curve positive samples, means that specific gene is amplified um, so specific gene is amplified. Here you can see the cycles at the X on the X scale. You can see the cycle 10 cycle, 20 cycle, 30 cycles and 40 cycle. So in the diagnostic we generally run up to the 39 or the 40 cycles, and the first sigmoid curve you will see around 18 cycles, means the viral gene was present in the abundance that it upscaled or it amplified at the 18th cycle, and hence it is called as the real-time PCR. Why? So here you can see it is amplified at the 18th cycle. Similarly, all other positive samples are amplified subsequently, means at the different cycles. And this is the CT value. And CT value means the actual value where the amplification of that particular gene started. So CT value 18th means at the 18th cycle that viral genome or the viral gene starts amplification. So more is the infection, it will pick up in the early cycles. Suppose here you can see the in the 18 cycles, even if in few cases we can see the amplification at the 10th or 12th run, 10th cycle, 10th or 12th cycle. It means that the abundance of the viral genome is that much that it get picked up in the 18th, sorry, in the 10th or the 12th cycle. So below, left hand side below diagram is the of the hex gene. It is again of the viral gene. So you can see the upper and below, both the what genes are showing the similar patterns because they are of the virus and they were in the abundance. On the right hand side, you can see the saffron colored RNSP. This RNSP is the internal control which is needed to find out whether the extraction is done properly or not. So if when the RNSP doesn't work, it means there is the issue with the sample collection or there is issue in the extraction system. So more the quantity of the gene, less will be the CT value. So in the viral COVID-19 diagnostic, we use up to the 30 cycle or the 30 or 32 CT values as a positive. Beyond that, we can consider it as a negative because sometimes it can show the false positive signal. Last one is the technique used in the detection of the proteins. Directly, it is not the technique used in the diagnostic, but after doing the SDS, we can do the Western blotting or we can use the mass spec analysis for the diagnostic purpose. But it is more common in the molecular biology labs. So how the protein is getting detected? So there are two types of what we can say polyacrylamide gel electrophoresis, or two types of pages are there. One is the native page and second one is the SDS page. Native means we use the protein in its tertiary structure, means or what is in the it is in the inside the cell. Means suppose protein will be active or the enzyme will only be active or it will perform its biological function when it is in the tertiary or quaternary structure. So in the native page, we use or we try to find out the native protein in its actual form. In the SDS page, we use the sodium dodecyl sulfate, that is SDS, that is the negatively charged detergent and it is the harsh detergent. So in the SDS page, we rupture all the structures, means like tertiary or the quaternary or even the secondary. So SDS page analysis is totally depend upon the sequence or the what we can say is the actual amino acid sequence. So native gel electrophoresis, it depends also upon the molecular weight and the radius. Means suppose the two proteins who are similar in their molecular weight but different in their spatial conformation or tertiary or quaternary structure, they can run differently. So the native page we can't use directly or it is not that much reliable as compared to the SDS page. So the SDS page is more common used in the labs So how SDS page works? So SDS is again anionic detergent. What it does, it ruptures the total structure of the protein and it allows the protein to run in the linear manner. And once it gets linear, it is totally overlapped with the SDS. So the total charge on the fragment or the amino acid sequence comes as a negative charge, irrespective of their molecular weights. So what are the components of the SDS page? So basically, we use the acrylamide and the bisacrylamide. So what is the acrylamide and bisacrylamide and what is its use? So basically, bisacryl and acrylamide, they form the actual what we can say the porous or the capillary structure. So the how it works, we use additional ammonium sulfate 10% and the timid. So in the presence of timid, the ammonium persulfate, the sulfate group of the ammonium persulfate gets separated. It works as the free radical. And this free radical or the SO4 minus charge gives the electrons to both these uh, monomers, like for to form the polymers. So bisacrylamide and acrylamide, actually they are inert, means they are non-reactive. But when they receive the electron from the ammonium persulfate, they cross polymerize, and you can see the pink color bisacrylamide forms the cross-linking between the polyacrylamides. So now the gel, similar like agarose, it is formed very thin slab. But the basic difference between the agarose gel and the SDS page is that agarose gel runs horizontally, but we run SDS page vertically. And in the SDS page, there are two different pages. Means the upper side is the stacking gel and the lower side is the resolving gel. So the stacking gel is with the different pH and the resolving gel is with different pH. Additionally, we use the tank buffer which is of pH 8.3. So in the total SDS based system, three different pHs are there. One for the stacking, one for the resolving, and one for the tank buffer. And why this much is necessary, we will discuss. Also, the total system is called discontinuous buffer system, and the reason is the same. In the agarose system, we use the TAE buffer with the same pH, or we use the single pH. In the discontinuous buffer system, we are using three different pHs. So why the stacking gel is used and why resolving gel is necessary? So stacking gel is with the pH 6.8. So at this pH, we use the acrylamide and bisacrylamide at the lower concentration. And hence at the stacking side, in the stacking gel, the porous structure will be a bigger as compared to the resolving gel. And what happens here? Here we load the proteins in the stacking gel. So you can see the different wells here. So in these wells, we add the proteins with the reducing buffer. So reducing buffer is again nothing but SDS and beta mercaptoethanol. SDS or beta mercaptoethanol, the principle of using it is just the rupture the tertiary or secondary structures of the proteins and make it linear. So how it works? In the buffer system, there is a presence of glycine. Glycine is the, again amino acid, and the buffer system is consist of tris, HCl, and the glycine. So the HCl or the chloride ions from the tris and the glycine are the main involved in the running of the gel. So how it runs? It's so similar. Glycine is actually negative charged in the pH 8.3 buffer system. So when it enters into the stacking gel, it acquires a little bit positive charge and it gets neutral. Once it get neutralized, it works as a deuteronic ion, means positive and negative, both the charges are there. And the buffer chloride from the buffer system, it runs faster than the glycine. So in between the glycine and the chloride ions, our proteins run. It is similar like in the front end, the chloride ions are there. In the back side, glycines are there. Glycine end is there, and in between, proteins are run during the stacking gel. When it enters into the resolving gel, the system get interchanged, means glycine again gets the negative charge as compared to the chloride ions, and it comes the forward in front, and the chloride ions they go into the back foot. And now again the due to the glycine, the porous size of the actual, what we can say, stacking resolving gel, it get narrowed down due to the glycine, because glycine is entering through the pore system. So the porous structure is again narrows down. So it is hard for the proteins to run through the resolving system. So what happens? The fragments, smaller fragments, run faster as compared to the larger fragments or the larger proteins. It is similar like in what we have seen in the agarose gel electrophoresis. The smaller DNA fragments run faster as compared to the larger DNA fragments. Same thing happens from the negative charge. Proteins run towards the positive charge anode. It is because of the SDS. Because due to the SDS, the charge on the fragments of the protein are the negative. So it starts running towards the positive charge. So this is the actual gel image where you can see the lower fragments or the lower proteins run faster, and the upper side are the larger bands or the larger proteins which run a little bit or slower as compared to the smaller fragments. So in the resolving gel, within the resolving gel, actual separation happens. In the stacking gels, we try to just bring all the proteins at the similar platform or the similar level. So SDS coated proteins, then here I have mentioned it is the linear molecules and charge remains uniform. and the fragments are run in this manner. So thank you. Hello.
Okay, sir. So, any doubt or queries? Any instruments to ask him? Okay. Thank you, sir,
Okay.
for delivering the talk on the tools of or the diagnostic instrument in molecular biology. Thank you, sir,
Okay.
for deliver the talk in different instrument. Deliver talk on the different instrument, and I'm also thankful to the uh, head of the department and vice principal of our institute, Professor Dr. V. Y. Deshpande, sir. I'm also thankful to the principal, Dr. B.T. Zadav, sir, for supporting and to the organizing like this faculty development program. And I'm also thankful to the all the faculty members to patients listening and attending this FDP. So, with the permission of organizer, I conclude the today's faculty development program. Thank you one and all.
Thank you. Thank you very much.